Welcome back, students. This is Module 4.1.2, Structure of DNA. It was in 1953 that James Watson and Francis Crick discovered the structure of DNA. They found that it was in the shape of a double helix. And for this discovery, they won the Nobel Prize. And they also, in the course of discovering the structure of DNA, they built a very famous model. Now, their discovery wasn't made in a vacuum. They, in fact, used data that was produced by a woman named Rosalind Franklin. She performed what is called X-ray diffraction. They'd actually crystallize the DNA molecules and then take X-rays of it. And that data was very critical in allowing Watson and Crick to figure out that it was a twisting ladder shape or a helix. Now, Rosalind Franklin, she contracted cancer and died without adequate recognition of her contribution. She is also often held up as a role model for women in science. She was treated poorly by the largely male scientific community at that time. But nevertheless, she lives on as an inspirational figure to women and all scientists. Here's a picture of Watson and Crick with their model. James Watson on the left, Francis Crick on the right. Watson was very young. He was about the age of some of you at this time. He was, I think, only 22 or 23 years old. And Francis Crick was a bit older. Of course, in America, we call it Watson and Crick. This work was actually conducted in Britain, and guess what they call the pair in Britain? Of course, they call them Crick and Watson. National pride knows a few limits. They do look kind of odd, and there's the Nobel Prize, which nowadays carries $1 million cash prize, a cash award with it. Here's a picture of DNA, the X-ray diffraction image of DNA, which, if you or I look at it, not being really experts in this kind of science, doesn't mean that much to us. Here's a picture of Rosalind Franklin, and DNA wants to know if that's its good side or not. The structure of DNA, the overall shape of the DNA molecule is a double helix. That's like a spiral staircase. You should be able to draw DNA. So here I'm going to show you. You should be able to draw it in several forms. To draw just an overall view of the double helix, you simply have to make a wavy line like so, then make another wavy line, and then connect the two with these little steps. That's not a very good version, but you get the idea. So that it looks something like that. Now, DNA we may define as a double-stranded nucleic acid molecule that contains genetic information and is capable of replicating itself. I note also here in the parentheses that it's a polymer of nucleotide monomers. And you should know that as well, that it is it fits the polymer monomer model very well, with DNA being the polymer, and the little building blocks or monomers are the nucleotides. And we're going to look at the structure of a nucleotide, and that will help us to understand the overall structure of DNA. So one nucleotide consists of three parts. There is the sugar, which we can see here. It's actually a 5-carbon sugar called deoxyribose. The phosphate group, which is here in kind of the yellowish. And then there's the sugar and the phosphate together. They make up the backbone on the outside of the ladder, and I'll show you that again in a moment. And then the third part, the kind of orange, is the nitrogenous base, and it's a basic molecule, and it has nitrogen in it. Hence, we call it a nitrogenous base. You should be able to draw how these fit together. So I'm going to draw it on the next slide. Here we have a blank slide, and I want to draw for you a nucleotide. So when you're drawing a nucleotide, you should draw the three parts. So I'm going to put an S here for the sugar. This is going to be have the phosphate group, and then out here we have the base. Now you should be able to draw several of them together. This is only one, and so I'm going to draw another here. There's our phosphate group. There is our sugar. And there's our base. Now, if we're going to draw more of them, the other ones are actually running in the other direction, upside down. So the B on the base is actually upside down. And so we can see our sugar here. And there's our sugar. It's upside down. There's our phosphate group. And so we can draw, again, another base. And these are actually connected by hydrogen bonds, either 2 or 3 hydrogen bonds. And we can draw, again, our sugar like so. There's our base and connected there. And of course, it'd be a phosphate group up here. And so you should be able to draw and indicate that this is, in fact, one nucleotide, and this shows 4 nucleotides together. And of course, if we're talking about the structure of DNA, remember we told you it's like a twisted ladder, or it's called a double helix. And so there's our ladder structure, and you can see the direct correspondence here, that the outside of the ladder is formed by the sugars and the phosphate groups. This is called the backbone here on the outside, going back and forth. And we can see there's 2 backbones. And then the rungs of the ladder right here are formed by these 2 bases. And of course, the ladder is in fact twisted. So we would see it like this. And so that's our double helix version. But remember, if I ask you to draw and show the nucleotides, you need to draw it like this here. So another fact to note about DNA is that the strands that make it up, that they run in opposite directions. So we actually have a term for that. We say that the 2 strands of DNA are anti-parallel, running in opposite directions. Now, we actually use numbering on DNA. So we could put here, for example, this is the 5' end, and this is the 3' end of DNA. While here, this would be the 5' end, and this would be the 3' end. Now, let me show you on this slide coming up here how we number DNA. It says right here that DNA is numbered 5' to 3'. Red is 5' to 3'. The prime is a little accent mark, if you will. And the numbers come from the carbons in the sugar. And so I'm going to make a little red dot, or actually I'll just number the carbons here. So that would be carbon number 1 at the corner right here, carbon number 2 at the corner, carbon number 3 right there, carbon number 4 right there, and there's a carbon right here, carbon number 5. And so you can see this is the 5' end, and this is the 3' end. So the reason that it's important to keep track of that, because beyond the fact that DNA is anti-parallel, this 3' end, it has a hydroxyl group, and this is where we can add new nucleotides. The 5' end we can't. So typically DNA grows on the 3' end, and so that's an important thing. And the reason we use prime is to separate from the carbons which are found over here in the base. And so there's carbons here too, but they don't have the little prime mark like these should. Okay, so that just gives you some insight as to where 5', 3' comes from. But I will expect you to understand what 5', 3' is, and in terms of anti-parallel, and in terms of adding new nucleotides to a growing DNA chain. So let's go on. So far, I've implied to you that the bases are all the same, but in fact, there are different types of bases. There are 4 types of nucleotides, depending upon which of 4 bases is present. And so the 4 bases are categorized under 2 headings. We have purines and pyrimidines. Purines are typically larger, purines are larger, and pyrimidines are smaller. And when the bases bond together, we always have one purine with one pyrimidine. In fact, these funny symbols that I've put here, it's meant to show that A always binds with T and G always binds with C. Now, I've just used the first letter of the purines and the pyrimidines, but you do need to know their proper names. The purines are adenine and guanine, and the pyrimidines are thymine and cytosine. And adenine always bonds with thymine, and guanine always binds with cytosine. Now, we do often abbreviate and say A and T and G and C. So A always bonds with T, and G always bonds with C. Important stuff to know. And so this bonding pattern is called complementary base pairing, and this is so very function as to underlie the very basis of DNA functioning. So do you think I want you to know that? You bet I do. You have to know A always pairs with T and G always pairs with C, and there's the the four showing their bonding patterns. Now, complementary base pairing was based upon the work of Erwin Chargaff. And here's his picture here, quite snappy with the bow tie, I suppose. He came up with what are called Chargaff's rules, and he was doing quantitative analysis of DNA from different sources. And he found that no matter what DNA he used, that the amount of A was always equal to the amount of T, and the amount of G was equal to the amount of C. And so that meant that the proportions of purines were equal to the proportions of pyrimidines. And of course, the purines were A and G, adenine and guanine, and the pyrimidines were thymine and cytosine. And remember, A always bonds with T, and G always bonds with C. And so this complementary base pairing explains what Chargaff found in his analysis. And that, in fact, gave rise, his analysis is in reality what gave rise to our understanding of complementary base pairing. And so just want to drive that home. This is a figure that allows us to review the basic structure of DNA. You can look at the molecule shape. It looks overall like a ladder. Remember, in real life, it would be twisted, so we'd call it a double helix. It has nucleotides as monomers. You should recognize one nucleotide right here. That would be one with its sugar deoxyribose, its base group, its phosphate group, and its base right here. And in this case, the base, it has a purine, and it has guanine there. Should note its directionality. Here's a 5' end. Here's a 3' end. Here we can see its anti-parallel. We see that this is a 3' end and the 5' end. And also note the complementary base pairing, G with C and A with T. And this actually shows these little dotted lines here represent hydrogen bonds, and there's 3 such hydrogen bonds between G and C and 2 hydrogen bonds between A and T. That's what holds the 2 halves of the ladder of DNA together. So this just gives you a little bit more insight into the structure of DNA. This is kind of the level of specificity that I'd like you to know about this particular molecule. It's a very important molecule, DNA, of course, as you know. So where is the information in DNA? As you can see on your screen, the information in DNA is stored in the unique order of the bases. So when we look at DNA from one species to the next, it all kind of looks the same. I mean, it has the backbone consisting of alternating sugar and phosphate groups. They're the same. And where we see differences, of course, is in the order of the bases, different letters of A, C, G, and T. In humans, there's over 3 billion such base pairs, and so it's that order of those bases that specifies a human being. One thing that I didn't mention previously that you ought to know, and that is that the backbone is connected with covalent bonds. They're actually given a special name because of the phosphate group that's involved. It's called a phosphodiester bond. But the most important thing to know here is that this is a covalent bond that holds the backbone together. So the backbone is very strong, while the bases are connected with hydrogen bonds, and so that makes them quite a bit weaker. And so you can unzip DNA, the two halves of DNA, where the backbone will stay intact, and then that will expose the information, because remember, the information is in the order of the bases, and by breaking the hydrogen bonds that hold it together, you can actually use each half of the DNA molecule as a template or something to copy, and so the information can be shared. So as I already mentioned, human DNA has over 3 billion base pairs. Four base pairs were represented previously. I drew them, and as well in the other diagram. And this is an important idea, is to understand overall, in a general introductory way, that DNA, the order of the bases, code for the orders of amino acids. So DNA codes for protein. It's that simple. So there we go. And so you might also wonder, how do you get a molecule that's not a protein? How do you code for that in DNA? Well, you code for a protein enzyme, and the enzyme directs the manufacture of other molecules, such as carbohydrates or lipids. This diagram is here just for your insight. I don't want you to memorize this, but this shows each atom in the different components of a nucleotide. So it shows here on the left, it shows the sugar deoxyribose right here. And so it shows all the carbons. And you already know that this is the 1 carbon, 2 carbon, I should say 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. But you don't really need to know its structure or anything like that. Then it shows a phosphate group here. So we have the sugar, the phosphate group. Those two are covalently bonded together with phosphodiester bonds to make up the backbone. And then you have one of several bases there. And you might be wondering about this other sugar and about this other base. Well, those are found in RNA, and we're going to study that in just a little bit here. But just want you to have this information. So with that, we're going to conclude this module.